Hello, this is Dr. Dan Guerra, come to you from Authentic Biochemistry Studios in the inland Pacific Northwest. Today, as you can see, is the 16th of April, 2021. It's taken a few days for me to decide whether or not I was going to do this particular video because I didn't want to leave it behind and not discuss this basic fatty acid metabolism lecture. And I was thought about doing it perhaps on just audio, but a lot of times people complain about that because it's difficult to follow, perhaps if you don't look at metabolic pathways. So I went ahead and I retooled the entire lecture so that I could give you a lot more very interesting information. Hopefully you will agree. So let's get started with this new and highly improved lecture. I don't know how you could really improve lipid metabolism, but I can do it because I'm a lipid biochemist. So obviously it will be improved. So let's just get right into this, shall we? So first of all, I'm going to talk to you about fatty acid utilization, and then we'll talk about biosynthesis. So free fatty acid can be generated from stored triacylglycerol. And that free fatty acid can go through a process known as beta oxidation, and then ultimately that carbon can be used for ketogenesis. This is all in the liver. Now, the triacylglycerol also has a glycerol molecule associated with it and three fatty acids, right? So that glycerol can also be run into metabolism, as shown here. An enzyme called glycerol kinase can convert it to glycerol 3-phosphate, and then the dehydrogenase, NADH producing, will make dihydroxyacetone phosphate, and then that will isomerize to glyceraldehyde 3-phosphate and be able to be used for glycolysis or even gluconeogenesis, right? So some of the carbon stored in triacylglycerol can be used for glucosynthesis, but not any of the carbon that's with fatty acid, because we don't have the right, the glyoxylate cycle. When I say right, I mean the appropriate pathway that plants and microbes have to be able to convert fatty acid carbon to carbohydrate. Mammals don't do that because we're missing two enzymes. And because of that, the fatty acids are used to generate ketone bodies. Right? That's the way that we mobilize that carbon. Okay. Now, in order to be able to get a fatty acid to be metabolized, it has to go from the cytoplasm to the matrix of the mitochondria. And it requires then, first of all, the fatty acid is esterified to coenzyme A, shown here. And then an enzyme that's in the cytosol, actually associated with the outer membrane of the mitochondrion, called carnitine palmitoyltransferase, will load that fatty acid onto this amino acid derivative called carnitine. That carnitine will be translocated with the fatty acid into the matrix of the mitochondria. And then a second reaction, catalyzed by carnitine palmitoyltransferase 2, will resynthesize the thioester. Now that fatty acid thioester, that fatty acyl group, can be passed through the beta oxidation pathway. Okay. And this is basically showing you the fundamental process. There's the fatty acyl-CoA. Now I want you to look a little bit here about the nomenclature. The alpha carbon is actually the second carbon in. The carbonyl group here is not called the alpha carbon, it's just called C1, right? So the alpha carbon in beta oxidation is actually the second carbon, okay? And so, however, because these two carbons will ultimately be derived to make acetyl-CoA, we call it beta oxidation because you're breaking the bond between the alpha carbon and the beta carbon. So that's why it's called that. So watch what happens. The first reaction is carried out by acyl-CoA dehydrogenase, which is FAD linked. And you synthesize flavin adenine dinucleotide reduced form. That will move through various electron transfer ferredoxin containing protein, ultimately to the ubiquinone oxidoreductase, and you will reduce it. So this is where the electrons are moving to a quinol pool and ultimately into the electron transport chain, which we've talked about at great length recently. And you can synthesize 2-ATP directly from that FADH, right? It has the electrochemical potential to generate 2-ATP from the very beginning of the beta oxidation pathway, okay? Now you're left with a trans delta 2, starting from the second carbon there. Enoyl-CoA, it's called enoyl because there's double bond here now. See, you've removed this hydrogen, right, from the beta carbon. Now, next reaction is a hydratase. So it's enoyl-CoA hydratase. You've added water over this bond. So there's your hydroxyl group, and you have an alcohol, right? This is 3L-hydroxyacyl-CoA. So now you meet up with the next dehydrogenase in the beta oxidation pathway. This is 3L-hydroxyacyl-CoA dehydrogenase, or HAD, and you synthesize NADH. So this is NAD bound to this enzyme, right? Now you've made beta-ketoacyl-CoA. You've completely oxidized this carbon here, you see? This beta carbon. It's completely oxidized. Now you can use coenzyme A, and this is called a thiolytic cleavage reaction. The enzyme is called beta-ketoacyl-CoA thiolase, and you've generated an acetyl-CoA and a fatty acid that's 2 carbons shorter. So beta oxidation goes through this exact series of reactions every time you make a new acetyl-CoA. So if you start off with a C16 fatty acid like palmitate, you're going to do this 7 times, right? And so you're going to generate that much acetyl-CoA, and all that acetyl-CoA ultimately then will be used to make ketone bodies, if that's indeed what's going on, ketogenesis, okay? Now, beta oxidation of fatty acids then provides FADH and NADH. And what that will do is shut down the TCA cycle, because the TCA cycle is functioning this way. Succinate dehydrogenase is bringing in FADH. It's going to use this reaction to synthesize FADH from FAD. So it will be blocked here. Then after the fumarase reaction, malate dehydrogenase, which is of course going to be generating NADH, that will be blocked because of the NADH synthesized from the beta oxidation. Now the other dehydrogenase enzymes in the pathway, isocitrate dehydrogenase, now ketoglutarate dehydrogenase, will also be inhibited because of the NADH made from beta oxidation of the fatty acids. So you're getting beta oxidation rather than TCA cycle. Okay. Now, here's where the actual interesting part of beta oxidation occurs. When I say it's interesting, it's because it gets a lot of popular press literature because people are inappropriately told that certain kinds of fatty acids will not move through beta oxidation. Particularly, people have been told that trans double-bonded fatty acids won't. I'm going to show you right now that that isn't the case, okay? Now, this particular fatty acid is linoleic acid. It's got 2 double bonds, so it's an 18-carbon fatty acid, and it's got a double bond in the 9 position, a double bond in the 12-carbon position, shown here. Now, these are both cis double bonds, cis versus trans in their geometric configuration. Okay. Now, double bonds where you have an odd and even carbon position, like here, that's odd 9 and even is 12. You have to overcome a couple of interesting biochemical events to be able to oxidize this fatty acid completely. And of course, the mammalian cell is able to do this without any problem at all. So starting with linoleic acid, we go through 2 rounds of beta oxidation like I just showed you, right? And you end up here going through the acyl-CoA dehydrogenase, and you make now because through that oxidation, remember we made that trans bond right there, right? So you have an intermediate called 2,5,8 trienoyl-CoA. Now, we'll meet up with the complete oxidation of this pathway, but you have a problem here because you've got a beta-gamma double bond, okay? Now, it's not a problem that you can't overcome. It's a problem if you are going to go through beta oxidation in a fashion that doesn't have that double bond positioning, such as a saturated fatty acid. So how does it overcome? Let's look. The beta-gamma double bond is an enoyl-CoA, and it's unsuitable as a substrate for the enyl-CoA hydratase. Okay. So an isomerase has to convert the cis delta 3 double bond, that's shown right there, to a stable ester conjugated trans delta 2 positional and geometrical isomer, right? So you've got this 3 carbon bond here, right? And you have to turn it into, 3 carbon, it's in a 3 carbon position. You have to put it into this delta 2 positional geometrical isomer. And that's what's right there. It's a trans double bond, okay? Now, if you have a trans fatty acid that you're carrying out the oxidation of, a polyunsaturated fatty acid that has trans double bonds, there's no problem because it already has the trans geometry, okay? So this isomerase will not be necessary to do that geometrical alteration. It will only cause the positional movement, right? Because the isomerase can do a cis-trans isomerization, which means it can also take a trans and turn it into a cis. Cis-trans, trans-cis. Freely reversible reactions. So once you have a trans double bond there, it will just already be directly able to go through the next round of reaction. Right? And so that's what you're going here is one round of beta oxidation, the first oxidation of the next round. So you're going to add the NAD, the FAD, the coenzyme A, the whole business, making the reduced forms of both those nucleotides and then a new acetyl-CoA. Now you're left with another, it's called a problem in this textbook, but it's not a problem. It just means that the beta oxidation process has to carry out yet another series of reactions to be able to process fatty acids that have multiple double bonds. Cis or trans, it doesn't matter. That's my point. So the next problem is that you are left with a 2,4, right, dienol, right, which is also not an acceptable substrate for the hydratase. That enzyme won't function with it. So now you have an NADPH dependent reductase. In fact, it's called the 2,4-dienoyl-CoA reductase, mammalian, not found in other animals. It's also not found in microbes. Okay, so that solves this problem of the hydratase not being able to function. Watch what it does. It does it by reducing the delta 4 double bond. Okay, that's exactly what's going to happen here. And it obtains a trans 3 double bond. Okay, and that's right there. Right? That's what you're going to end up with. So you have now this NADPH dependent reductase. So you had this isomerase, and now you have this reductase to be able to process polyunsaturated fatty acids. Okay. Now that you've made this trans 3 double bond, which is right there in red, so, and it's called 3,2-enoyl-CoA isomerase. Now this enzyme, right, it will allow for the completion of the oxidation. So you've had an enoyl-CoA isomerase, you've had a 2,4-dienoyl-CoA reductase, and you've had one more enzyme necessary to complete this cycle, a 3,2-enoyl-CoA isomerase. Now you're back to the correct position. You can do the hydration, right? And the continuation of the beta oxidation. Now, I want you to also notice something else. There's also a problem with the 3,5,8 trienoyl-CoA coming from the 2,5,8. You've got the trans double bond here between 3 and 4, and 2 cis double bonds, the 5 carbon and the 8 carbon. Okay. The problem here is having a 2,5 dienoyl-CoA to a 3,5 dienoyl-CoA product, right? So you had 2,5. We're not even talking about the 8 here because this is the business end of what's going on, right? But now you've got a 3,5, right? So you had a 2,5, now you have a 3,5. After this isomerase, the same isomerase that functioned down here. Okay. Now, that's left by that 3,2 isomerase, as I just said, and it's corrected by a double isomerase called the 3,5,2,4, okay, the 3,5,2,4 dienoyl, okay, coenzyme A isomerase, which gives us the 2,4,8, right, the 2,4,8 trienoyl-CoA. Right, when you have now 2 trans double bonds. Now keep in mind, this is going to be functioning the same thing with a trans fatty acid, only you won't require the isomerization, only the position will shift. The enzyme does both. Okay? So it has absolutely no hindrance of these reactions having trans fatty acids. No hindrance whatsoever. Right? So you ultimately then have the 2,4-dienoyl-CoA reductase, and the next step is facilitated again by the 3,2-dienoyl isomerase, which I already described to you. That was the one that was down here. Okay. Same one that ended up with giving us, quote, a problem, which is fixed by this 3,5,2,4-dienoyl-CoA isomerase, giving you the 2,4,8 trienoyl-CoA, meeting up with the 2,4-dienoyl-CoA reductase, again the NADPH, the same enzyme that's here, giving you now the 3,2-enoyl and the 3,2-enoyl-CoA isomerase then functioning, carrying out the rest of the beta oxidation. Right? So this is how all fatty acids with double bonds, multiple double bonds, are beta oxidized. Right? I showed you linoleic acid. It could be alpha-linolenic acid. It could be icosapentaenoic acid. It could be docosahexaenoic acid. It could be any of those fatty acids with trans double bonds. They'll all go down the same beta oxidation pathway without any halting, without any interruption of the pathway. So trans fatty acids that you find in the diet, particularly they can be synthesized by the rumen in ruminant animals, both in fatty acids that are found in meat, but also in dairy products. So naturally occurring fatty acids in dairy products and in meat to have some trans double bonds because of the hydrogenation reactions that occur in the rumen of ruminant animals, where we get a lot of meat and where we, of course, get dairy products. Okay? So having trans fatty acids in the diet is nothing unusual for mammals. Right? That's the other point I want to make. Nothing at all unusual. All right. So I hope that made that clear for you now. I've given you each of the enzymes necessary to carry out the complete beta oxidation of a polyunsaturated fatty acid, whether it be all cis bonds or cis trans bonds. It's not at all a problem because of these exact enzymes, these isomerases, and the reductases that will move positionally those double bonds so you can carry out that hydration and finish the beta oxidation process. Okay. Now, talk to you about something else. Talk to you about conjugated double bonds. So a double carbon bond conjugation, sensu stricto defined, is where you have pi electrons, that is, electrons in the p orbitals, and they can be shared over more than 3 or more carbon atoms. Okay. So this molecule here has adjacent double bonds, right? Right? Because that carbon there is shared with this carbon. There's a double bond there. There's a double bond right with this carbon here, right? It's what we call a conjugated diene, two double bonds. Now, you didn't have those conjugated dienes in the fatty acids we just ran through the beta oxidation pathway. So this is an unusual thing. Now, this, what we normally have is what we just saw, normally meaning fatty acids that we metabolize, cis or trans double bonds, doesn't matter. That kind of configuration is shown here. Positional geometry shown here. Doesn't have adjacent double bonds, it isn't conjugated. Now, there are other ways of understanding this conjugation. First glance, none of these molecules look like, because they seem to appear like this. They don't seem to be conjugated, but they each are. So this has nothing to do with the trans configuration. It has to do with the electrochemistry. So these alkenes are all alkenes. One is a carbocation, one is a carbanion. One of them has a heteroatom, an oxygen atom, and one of them is a radical. All of those are still conjugated systems. Okay? So those are the four classes. They participate in what is known as electron resonance isomery. And there are four classical cases, as I just said. Carbon or other atom with an empty p orbital, or a carbocation. A carbon with a half-filled p orbital, or a radical, shown here, right? An unpaired electron. That's what a radical is in chemistry. A carbon with a lone pair of electrons, that's a carbanion. And then any other atom in the lone pair, I showed you oxygen here as a atomic structure within the fatty acid backbone. In each of those, you can draw resonance forms where the middle atom participates in that pi bond, that p orbital, and that implies that the electrons are being shared or delocalized. Same kind of isomery happens actually in ring-structured compounds. And that's between the central atom and the alkenes on either side, and thus the whole system is considered conjugated. Okay. So I wanted you to know about conjugate double bonds so that you understand what those are, because I'm going to tell you something else a little bit more interesting and into the heavy level of sophistication of lipid metabolism. Okay. I can only figure out how to make my cursor get out of the way of that so I can. All right, here we go. Sorry about that. Oh, now we got to go back. Here's a paper published in the Journal of Nutritional Biochemistry that I'm going to talk to you about briefly right now. I guess I'm going to leave myself there. The alpha-3 Ileosteric acid, or alpha ESA, is a cis-9 trans-11 trans-13 isomer of conjugated linolenic acid. And you find it in something called bitter melon seed oil. It's naturally occurring. Okay. Trans fatty acid with a conjugated system. Okay. In hepatoma cell lines, this paper is going to tell us, this alpha Ileosteric acid significantly lowered intracellular triacylglycerol accumulation compared to using a control fatty acid, which is alpha linolenic acid. That's a 9-12-15. Those are non-conjugated double bonds. And those are all cis. Now, this alpha ESA lowered TAG accumulation in a dose and time dependent manner. That's really interesting. Now, why are they using this conjugated fatty acid? They're using it to try to control steatohepatitis and steatosis in people that have fatty liver. Okay? That's why it's published in the Journal of Nutritional Biochemistry. So the alpha ESA enhanced actually what else it was doing, because it decreased triacylglycerol accumulation. It enhanced beta oxidation, what I just went through for you. And it lowered, therefore, lipogenesis, including the synthesis of the major storage depot fat triacylglycerol. So alpha ESA, this paper described for us, what it did when you read the entire paper, is it triggered AMP protein kinase, AMP kinase, activation, but it didn't seem to alter sirtuin-1 protein levels. Now, why is sirtuin involved? Because sirtuin, I think I mentioned to you many times recently, has a direct modifying effect on levels of NAD and NADH. In fact, sirtuin is activated by binding to NAD+, the oxidized form of nicotinamide adenine dinucleotide. So watch what's going on here. It's very interesting. So it triggered this AMP kinase without altering sirtuin-1 protein levels, which doesn't surprise me because, let's continue now, with alpha-linolenic acid alone at 100 micromole per liter, or in combination with alpha ESA, shown in those concentrations there, for 24 hours, the acetylation of FOXO1, which is a transcription factor, okay, was decreased, suggesting that SIRT1 was activated. I just told you the SIRT1 protein levels didn't change, yet alpha ESA had this effect. So how did it work? Here's how it worked. NAD to NADH ratios, messenger RNA levels for the nicotinamide phosphoribosyltransferase transferase, which I've talked about recently when we talked about sirtuin metabolism, were all increased when you had alpha-linolenic acid plus alpha ESA treatment compared to treatment with the normal alpha-linolenic acid alone. So the conjugated fatty acid did these things. It made the ratio of NAD to NADH higher, which means it activated the sirtuin. Right? It also increased the levels of NMAMPT, which constantly recycles nicotinamide adenine dinucleotide for sirtuin, but also this other enzyme called PTGR1. Now, it increased the transcription of those two because it was altering FOXO1 acetylation, because the sirtuins are a deacetylase, right? Okay, so you see how this works. Very interesting, right? So it suggests that the alpha ESA, this conjugated fatty acid, which you find in the bitter melon, right, which is going to be used now as a drug to combat steatosis, and not only fatty liver, but also steatohepatitis and possibly hepatocellular carcinoma, which ultimately can be caused by a fatty liver, non-alcoholic fatty liver, the NAFLD type that I've talked about. Okay, suggests that alpha ESA activates SIRT1 by increasing NAD synthesis. One of the reasons is because of that NAMP2 pathway. And it also takes NADPH and consumes it. Now, what does that have to do with something? With anything here? I'll explain it to you in some detail. You'll see. The anti-steatosis effect of alpha ESA we've been discussing here from this paper was confirmed in mice treated with a high sucrose diet, which will induce high levels of triacylglycerol, sugar converted to triacylglycerol, supplemented with 1% alpha ESA for 5 weeks. This is a nutritional study. It appears, therefore, that alpha ESA favorably affects hepatic lipid metabolism favorably by increasing cellular NAD to NADH ratios and activating at the same time transcription factors and kinases. So transcription factor PPAR alpha, which is involved in transcription of beta oxidation genes, transcription and then ultimately, presumably, translation, so higher levels of beta oxidation. All right? The AMP kinase, which also induces the process of beta oxidation, and the SIRT1 cycling pathway because of the increased levels of NAD. All of it explained now. Now, I'm going to go back, because this is authentic biochemistry, and say, what about this PTGR1? Now, that particular enzyme involves this leukotriene metabolism, inactivation of a chemotactic factor, leukotriene, which is an oxygenated polyunsaturated fatty acid, an icosanoid actually. Inactivation of the chemotactic factor, leukotriene B4, this PTGR1 catalyzes the NADP-dependent conversion of leukotriene B4 to 12-oxoleukotriene B4. Okay? Thus, inactivating the chemotactic factor, right? Which can result in downstream processing of inflammation, ultimately potentially leading to hepatocellular carcinoma. Now, let's take a look at this in some detail. Now, first of all, because this is biochemistry, we have to tell you what the enzyme does. That's what I'm doing here. Take 13,14-dihydro-15-oxoprostaglandin E1 and NADP, the oxidized form of nicotinamide dinucleotide phosphoryl group, modified, to generate 15-oxoprostaglandin E1 plus a proton plus reduced NADPH. Now, this enzyme, PTGR1, catalyzes with high efficiency the reduction of the 13-14 double bond of 5-oxoprostaglandins, including 5-oxo-PGE1, 15-oxo-PGE2, 15-oxo-PGF1 alpha, and 15-oxo-PGF2 alpha. So the reaction actually works in the reverse of what's shown there, right? So that means it consumes NADPH. That's why it was significant the transcription of this gene went up, because ultimately it looks like the enzyme activity also increased and therefore reduced the levels of NADPH. All the while controlling the level of the chemotactic icosanoid, 15-oxoprostaglandin, right? Which ultimately, this is the prostaglandin pathway, ultimately will also control the leukotriene B4, right? Because it's carrying out the same reaction, right? The reverse here. Thus removing this chemotactic agent, thus ameliorating steatosis, right? Fatty liver. All of that positive, all controlled by that conjugated fatty acid derivative that you get from the bitter melon. Okay. Got it? Now, we're not done. From this GeneCards site, I was able to look at where this PTGR1 is expressed normally. You can see it's bone marrow, white blood cells, in the brain, the small intestine, various other organs, spleen, stomach, esophagus, bladder, pancreas. It's in the salivary gland. It's also in the skin, and it's also in a lot of reproductive tissues: ovary, uterus, placenta, prostate, and testes. Okay, so this is RNA sequence data. This is a microRNA data showing you where that transcript is found in multiple tissues. Immune cells, nervous system, particularly central nervous system, skeletal muscle, internally, all those different organs, secretory pathways, and reproductive. Okay. Now, I also wanted to show you where the protein is found. Okay. And you find a lot of the protein in peripheral blood nucleocytes, at the lymph node, tonsil, bone marrow stromal cells. A lot of protein also shows up in the central nervous system, including the spinal cord, the retina, cerebral cortex, the prefrontal cortex. A whole series of other organ systems, the protein is found: the colon, the rectum, the liver. Of course, it's important from that last paper. And ultimately also in a lot of reproductive systems: cervix, ovary, testes, again, okay, and the seminal vesicles. So you get the idea where the protein's expressed. Now, I just talked about the one in the liver. Now, if you're modifying the activity because of the expression of that enzyme, right, the one that's going to convert that leukotriene B4 and therefore remove the chemotactic factor, what's that going to do overall by increasing the amount of that conjugated fatty acid, that alpha ESA? could have a tremendous pleiotropic effect on the body, even though it might actually be useful for controlling steatohepatitis. See? But there could be downstream off-label activities, because I wanted to look at where else you found this enzyme. A lot of places. That's important. It's very important. Now here's a paper, Free Radical Biology in Medicine, published in January 2017, which shows this significance and shows why you're listening to authentic biochemistry. So that prostaglandin reductase, the PTGR1 I just talked about, is of course an alkenal, that's an aldehyde, or an alkenone, right? That's the ketone, oxidoreductase, that is involved in the catabolism of those eicosanols I just showed you, and the lipid peroxidation pathways such as 4-hydroxynonenal, which we talked about a month ago here in the Authentic Biochemistry series. So this enzyme, PTGR1, is overexpressed in human, clinical, and experimentally induced samples of hepatocellular carcinoma. Oh, interesting, right? So there's a normal hepatocyte. It's considered an oxidized cell with the membrane showing a status of about 250 millivolt. Remember that when we talked about the membrane potential, right? I always want you to remember all the lectures I gave you. And this 4-HNE, and of course hydrogen peroxide, is going to introduce, it's going to cause oxidative stress, and it can cause actual cell destruction, right? Cell death, usually from a programmed cell death pathway because of HNE and hydrogen peroxide. Now, we can ask the question, what's the link of the induction of PTGR1 expression by the activation of nuclear factor, that's the erythroid-derived 2, like NRF2? Okay. PTGR1 was overexpressed during early phase in altered hepatocyte foci. High-level expression was maintained in persistent nodules until tumors developed. Okay? So PTGR1 expression was regulated by NRF2, which we talked about before. NRF2 is also found in immune cells. That transcription factor is. And it's bound to an antioxidant response element at exactly minus 653 base pairs in upstream part of the promoter region of PTGR1 gene. The activation, therefore, of NRF2 induced the activation of the antioxidant response that included effects on proteins such as the glutamate cysteine ligase, which we talked about during our amino acid catabolism lectures, and catalytic subunit NADPH dehydrogenase quinone 1, which we also have talked about, and of course the glutathione S transferase GSTP1. All of these effects cumulatively may have pronounced reduced status that was associated with a high proliferation rate in these tumors. Now, if PTGR1 is stably expressed, there is a reduction time required for the proliferation and protective effect against hydrogen peroxide and 4-HNE induced cell death. So all of this suggests in the tumor cell, you see, because it's blocking this enzyme is going to be blocking, it's going to cause high proliferation rate, right? And it's going to evade cell death when you have a lot of this protein transcribed from the NRF2 transcription factor. That's what this paper is talking about here. So, suggests that PTGR1 and antioxidant responses act as a metabolic adaptation in tumor cells and can in fact contribute to proliferation and cell death evasion in those liver tumor cells. Now, I just told you that conjugated fatty acid, right, is controlling the activity of that enzyme. And they thought that was good in that paper, that nutrition paper, biochemical nutrition paper, because they were looking at steatohepatitis. But if you look at tumor cells themselves, high-level expression of that protein, which was induced by that conjugated fatty acid, can actually decrease cell death and increase tumor proliferation. 1 year later, a different paper entirely. This is why it's really important to think about any potential pharmaceuticals as being touted to being useful in some neoplasia or in some pathophysiological system, such as steatohepatitis, but actually upon closer examination could induce more tumorigenesis. That's why it's important to read all the literature, right? That's why I always say in authentic biochemistry, you cannot come up with a single locus of understanding until you understand what the literature, the published peer-reviewed literature tells us. Before you start running to think that a conjugated fatty acid like alpha ESA could be useful to control cancer. It can actually cause cancer proliferation because that PTGR, that one transcript that's being generated from that FOXO1, because of that acetylation pattern and the NAD to NADH ratios I just talked about. Now, you wouldn't know that if you didn't know these two papers. I know these two papers, and now I explain it to you, and now you understand why it controls beta oxidation, and yet it can have a bad effect on tumor proliferation, it being this conjugated fatty acid. Now, I want to show you, just so that you have this in your records, here are some of the trans fatty acids you find in foods. These can be hydrogenation products because there is hydrogenation from the microbes in rumen. So you're going to get these fatty acids in dairy or in chemical processing, partial hydrogenation of fatty acids. Now, the reason they carry out partial hydrogenation of fatty acids and generate some of these trans fatty acids is to control the level of polyunsaturation in foods. Now, why was the food industry doing this? It was doing it to control rancidity because of auto-oxidation of stored food. That's why chemical hydrogenation was being performed. Now, with high levels now of oleic acid, monounsaturated fatty acid, increased in the diet, you have less and less of the polyunsaturated fatty acids. But it also means you can get an inappropriate omega-6 to omega-3 fatty acid ratio, which can lead to cardiovascular disease, right? Because we've lowered the amount of polyunsaturated fatty acids in the diet. Now, you see, once again, you end up with an antinomy. Antinomy means you have the possibility of confirming a proposition that could be completely contradictory. So the antinomus, means against the law of logic, right? You can both prove and disprove one proposition, right? A or not A can both be proven. That's an antinomy in logic, right? Antinomos, against the law, the middle term law. And why am I telling you this? Because that is actually how you must understand research science, not just biochemistry, but pathophysiology, molecular genetics, epigenetics, and everything that's in biology. Because that's, at least as far as I'm willing to take it, but in all of research science, you have to have the complete understanding of these pathways, complete understanding of all the cell types, for you to come up with a comprehensive understanding of health and of pathology. So if you go about listening to popular press journals and you think you understand something about nutrition and exercise and even pharmaceutical drugs, or that you and your physician know the ins and outs of a given pharmaceutical to control, say, type 2 diabetes or metabolic syndrome or obesity or cardiovascular disease or the potentiation of carcinogenesis. Be very careful to understand that in research science, we are at the level where we do comprehend that there can be completely contradictory results with a given pharmaceutical or dietary intervention. And you have to understand that before you lead yourself down a pathway that can corrupt what you think you're actually trying to preserve. That's the point of being a biochemist and listening to a biochemist, an authentic biochemist, explain to you what's in the scientific literature. Once again, these fatty acids that have trans double bonds, because they're not conjugated, you can see those are not conjugated. None of these are conjugated double bond systems. And that trans double bond will have absolutely no effect on the normal beta oxidation pathway. Therefore, the fatty acid will be metabolized and will in no way affect any of the transcription factors or any of the potential pathophysiology that is normally ascribed to dietary trans double bonded fatty acids. This is why the trans fatty acid story is largely debunked if you look at the scientific literature, not just at the result of using scientific literature inappropriately in certain medical studies, it's particularly when they're done with model animals, like with mouse or with rat models. Okay, so what happens with rat and mouse model studies in trans fatty acids is not the same that's occurring with trans fatty acids in the normal physiological response in humans. And this work was well studied. Trans fatty acids in humans was well studied in the 1980s by a research group in the Agricultural Research Service in Peoria, Illinois, where your professor here actually did a postdoctoral fellowship. Right? And I worked with the people. I wasn't studying trans fatty acids at the time, but my research leader in his group was. And they published many, dozens and dozens of papers on GC mass spectral analysis of the metabolism of trans fatty acids in medical students. They were being given deuterated fats, so they were using D2 just to follow, because of mass spectroscopy, the metabolism of trans fatty acids in these humans, and they had no ill effect on the people. And in fact, they also were able to show in numerous studies, like I said, dozens of papers, very well done papers from Ed Emkin's group and Rohiter's group. And you go back and look at those papers in Chemistry and Physics of Lipids, for example, and in various other journals too. JPC is another one they publish in quite extensively. The trans fatty acids had no deleterious effect on humans. Okay, just want to make that clear to you. Now, if you have an odd-chain fatty acid, you end up with propanyl-CoA. That's obviously a 3-carbon fatty acid, but watch what happens to it. Propanyl-CoA carboxylase will make methylmalonyl-CoA. Methylmalonyl-CoA racemase will alter the structure. We'll put this carboxylic acid on this carbon. Now you have R-methylmalonyl-CoA. And then a simple mutase reaction moving around, these substituents will generate succinyl-CoA. And now succinyl-CoA will run right through the TCA cycle. So odd-chain fatty acids can be converted because of the synthesis of propanyl-CoA at the end, right? Because it's odd-chain, as you end up with 3-carbon, not 2. You can convert a succinyl-CoA and run through the TCA cycles. That's odd-chain fatty acids, which are also found in dairy products, can be completely metabolized without any problem with normal metabolism. That leads us finally to what's left over with even chain fatty acids and ultimately making ketone bodies. And there they are: acetoacetate, beta-hydroxybutyrate, which is the alcohol, and then acetone is kind of a byproduct. It's not really utilized in ketogenesis because it's a volatile compound. You do smell it on the breath of people that are ketogenic. Right? And in diabetics, there's much more acetone. And some of it does get moved through metabolism, but very little of it can be metabolized any further because it's a volatile. So very low amounts of acetone are actually generated. The two main ketone bodies, of course, are always acetoacetate and beta-hydroxybutyrate, which are interconvertible with an NAD-mediated dehydrogenase. So that leads us then to where I'm about ready to end here, because we did the beta oxidation pathway, and I really wanted to do the trans fatty acids and the conjugated fatty acids. And then that led us into conjugated fatty acids having this antimonic effect in human disease, right? Antinomy, remember? So we're just going to end here in straight form. Fatty acids and amino acids can be converted to acetyl-CoA. There's an enzyme called ketothiolase. You take 2 acetyl-CoAs, you make acetyl-acetyl-CoA. There's an enzyme that follows called HMG-CoA synthase. This is all in the liver mitochondria, which takes another acetyl-CoA that makes the 6-carbon hydroxymethyl-glutarate-CoA, which is then converted to acetyl-CoA and acetoacetate, which is, of course, a ketone body. And that's carried out by the enzyme HMG-CoA lyase. The acetoacetate, as I just told you, with NADH being converted to NAD, will because you're going to reduce this now, you're going to reduce acetoacetate to the beta hydroxybutyrate. So those are your 2 ketone bodies. Now, they are freely water-soluble carbon compounds. They make it through circulation, and then they end up in the mitochondria of muscle or the brain or any other tissue, really. And beta hydroxybutyrate is reconverted to acetoacetate, now with the same enzyme, beta hydroxybutyrate dehydrogenase. So now you are actually converting NADH to NAD, so you're using up some of the NADH. Acetoacetate then through an enzyme called thiophorase will react with succinyl-CoA to make succinate, but transferring now the CoA, right? And now you have acetoacetyl-CoA, and the ketothiolase reaction, the same one we saw back here, will give you 2 acetyl-CoAs, and they can be run right through the TCA cycle. Okay. So now you see the biosynthesis of ketone bodies and then your utilization. When you run low on glucose in circulation, you're also going to be gluconeogenic here, but you can convert some of that depot fat then directly to ketone bodies. This is the whole point I'm making to you today. Okay? Beta oxidation pathway making NADH and FADH2, they can drive gluconeogenesis during the fasting cycle or the starve cycle after glycogen reserves are knocked down and free circulating glucose levels drop and the glucagon activity increases from the pancreas, the hormone from the pancreas, generating hormone-sensitive lipase-mediated movement of fatty acids from storage depot to the liver to carry out these reactions to then generate the NADH and the FADH2 to run gluconeogenesis, right, from non-carbohydrate, non-fatty acid precursors, but from the beta oxidation of fatty acids, giving you the reducing power to make the ATP necessary to run the gluconeogenesis. I always say this, right? But also to make these ketone bodies. All right, so this is just showing you the same reactions with the structures in place, but we're not going to do that right now. We're going to stop here because I think we went through enough of our, I think, very well described now beta oxidation pathway. You understand it. You understand how to deal with polyunsaturated fatty acids because those unusual isomerases and reductases, unusual only because you find them in mammals and not in other, you don't find them in microorganisms, higher plants to any significant degree, or in other animal populations besides mammals. Mammals have no problems dealing with polyunsaturated fatty acids. In fact, 2 essential fatty acids, linoleic, I showed you that oxidation. Alpha-linolenic, I showed you that too. But also any fatty acids that we acquire in the diet that have transable bonds, because those are not conjugated systems. Then I went through the conjugated pathway, showed you what conjugated fatty acids can do in terms of transcriptional activity and ultimately post-translational modification of activity of enzymes, such as the sirtuins, then controlling transcription factors and turning on the expression of genes like the PTGR, which ultimately can down-regulate apoptosis and allow for cellular proliferation in the same liver that had been intoxicated, perhaps in early stages of pathology, because of increases in storage lipid in the form of triacylglycerol. Okay, so we're going to stop here. I think we are about ready to sign off. So again, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios on the 16th of April, 2021. Hopefully you enjoyed this lecture. I know I enjoyed doing it because it's lipid metabolism. I especially like doing it because we actually got to talk a little bit about some stereochemistry, which I also enjoy immensely, and I hope you did too. So Friday afternoon, enjoy yourself, and we will be back with you soon, either with a video lecture or more likely I'll go to the audio versions of Authentic Biochemistry to move back up to where we want to get back ultimately to the dialectical event ontology of aging, the immune system, epigenetics, resulting in morbidity and ultimately mortality in humans. So this is Dr. Dan Guerra from Authentic Biochemistry Studio saying bye for now.